Lab Practical 1 review of Lab 2. In this lab, we did several different short activities, but kind of all added up together to make one lab. One of the things that you should be familiar with are the differences between longitudinal sections and a cross section. If you see a picture or a slide, if we have our object in a long view, then we have a longitudinal section. If we have more of a round shape, then typically we're going to be seeing a cross-sectional view. Longitudinal section is going to show us the whole cell at one time, as if we were looking at the cell from the side, whereas a cross-section is going to show us a view as if we had cut through the cell or the object and we're looking at that object from the top down inside from that direction. Another topic we covered in Lab 2 was just a very general overview of the metric system. We did not do any metric conversions, so you will not need to be familiar with anything that I have marked in Table 2.1. But we did talk about length. The basic unit for length in the metric system is the meter, and we had various derivations of that: centimeters, millimeters, nanometers. On page 19, we talked about the metric units for weight or mass, gram, milligram. When we use a balance, we typically measure in milligrams. If you're doing other metric conversions, you might measure in kilograms, like the weight of a human body or something like that. We talked about a couple of different ways to measure volume. If we're measuring the volume of liquid, we typically use liters or milliliters, some derivation of that. If we're talking about the volume of a solid, then we're going to use length times width times depth, and we come up with a cubic measurement. Could be centimeters cubed, meters cubed, millimeters cubed, some derivation of the meter. When we measure a liquid in something like a graduated cylinder, we want to measure using the meniscus. And we want to be sure that we measure at the bottom of the meniscus, and we want to be sure that we're at proper height. We want to get down and look at the meniscus at eye level to be sure that we have the most accurate measurement. On page 21, we talked about temperature, and that there is the English unit Fahrenheit, which is very common here in the United States, but the rest of the world is going to use the metric unit Celsius when it's measuring temperature. So those were our discussions about the metric system. We applied that to some things that we were measuring on the microscope. And speaking of microscopes, on pages 22 and 23, you have a discussion about the differences between light microscopes and electron microscopes. Here on page 22, we see some discussion about the difference between the stereo or the dissecting microscope, which we would use to view a larger object like a whole fly, a piece of bark, lichen, various objects, versus the compound light microscope, which is going to measure very small materials. It's called the compound light microscope because it has two places where it measures at the ocular or the eyepiece and the objective that we have clicked into place. We talked about electron microscopes on page 23. There are two types of electron microscopes. We have our transmission electron microscope. Capital T, capital E, capital M is the abbreviation for the transmission electron microscope. It's going to view an object in the middle, the center of the object. We're going to have that object sliced very thinly, and we'll be able to transmit electrons up through that object. The electrons are received by a computer, turns it into an image of what we see inside the cell or other object. A scanning electron microscope is going to have electron beams shooting at the outside of the object, and we're going to be able to see the surface of that object. Electron microscopes have a much greater resolving power. They're able to show us very, very small distances between objects, nanometers of distance between two objects, and then we'll still be able to see those objects distinctly from each other and very clearly most of the time. Electron microscopes can measure up to 1,000,000 times, whereas the light microscopes, the ones we use in class, measure up to 1,000 times. They could measure as high as 2,000 times. We do not have that capability. Our microscopes only measure to 1,000 times. We skip on over to page 26. We have some rules about microscope use and care, so you might want to look those over. We try to practice those every time we use the microscope. On pages 26 and 27, we went through the parts of the microscope. So you should know the parts of the microscope by what the structures do, what's their function, as well as you should be able to label the parts of the microscope. You should be able to correctly identify the parts of the microscope. Most of the time I have real microscopes sitting out, have a large arrow pointing to a specific part of the microscope, and you're going to find the microscope part on a list of the multiple choice selections and pick the answer that best matches where the arrow is located. So be sure you know the parts of the microscope by structure, what they look like, where they're located, and then also by function. What does each part of the microscope do for us? On page 28, we talked about the fact that on a microscope our images show inversion. All the images that we see through the compound light microscope are going to be upside down and backwards. We talked about the fact that our microscopes are parfocal, which means once we get the microscope's slides focused, when we go to the next higher power, we're pretty much focused. We're going to have to do some adjustment, but it's actually a very small amount of adjustment since mostly our microscope slides are going to be focused. Once we get them focused on a lower power, go into a higher power, we should still be focused. That's parfocal. On page 29, we discussed how to calculate total magnification. We filled out this chart about the ocular lens times the objective lens and how that gives us total magnification. We talked about the fact that the scanning power gives us a total magnification of 4x. Low power, which is usually marked by the yellow ring, gives us a magnifying power of 10. So when we have the eyepiece at 10x and the low power objective at 10x, that's where we get the number 100. 10 times 10 is 100 for the total magnification when we're on low power. High power on our microscopes is marked with a blue ring, and that's each high power objective magnifies 40 times for a total magnification of 400x. Our oil immersion, which has a white ring around it, gives us a total magnification of 1000. On page 31, let me go back to page 30. On page 30, we did not talk about depth of field. So back to page 31. I was right the first time. So on page 31, we talked about how to make a wet mount slide, which we did with some onion cells. I think we did that with some cheek cells at one point. If not, we'll get there. But that's what we talked about, and cheek cells looked kind of like this if we did that. And so that's what we talked about in Lab 2. Some of the very basics about the metric system and basics of the microscope and how to use the microscope.